Thank you, sir.
Thank you. Excuse me. Okay, are you able to see the slides?
Yeah.
Yes, sir. We can.
Okay. Okay, just let me check how many people have joined, then we can start.
I mean, anytime,
Okay, people have joined now, so we can start the talk. So, Divya, are you ready? No?
Yes, sir. I'm ready.
Okay, so I'll just put the slides. So, very good morning to all of you, and good afternoon, people who are joining from other parts of the east side of the country, and maybe good evening for those who have joined from the west side. So this is Air Quality Management lecture series. Today we are celebrating 70th lecture. We started more than 5 years back, and thanks to Dr. Divya for kindly agreed to give this month's talk. As I mentioned, we started this air quality management lecture series in October 2020 during Corona period, and it is one of its kind, not only in India, maybe across the globe, where this provide a platform for young researchers, scientists, engineers, educators, practitioners to share the latest advancement in theories and applications related to air quality. So lecture includes recent developments, innovations, use of recent technologies, and monitoring and analysis. So all which is related to air quality management. So it also provides platform for collaboration with stakeholders and experts from various countries. So these are all our past speakers, and if you can look at it, we could able to reach out to all the top experts across the globe who are working in air quality related areas. So, and you will see there are 70 photos will be there, and including today's Divya's photo. as a part of this air quality management lecture series, we also started an air quality management association. And the idea is to enhance understanding of the science behind air quality and promoting the knowledge of clean air to the local community. So we have various issues we have decided to address: ambient air quality, indoor air quality, health risk, and technological interventions and policy formulations in addressing the any topics related to air quality. AQML also provides a common forum to the interact with one another for mutual benefits. And if you are interested, all of you can become a member for this Air Quality Management Association. And we also started this Indian International Conference on Air Quality Management again 10 years back. So now we have 11th Indian International Conference on Air Quality Management. We're going to held on IIT Madras in December 14th to 18th. So please visit more details in this particular website. So I welcome Dr. Divyavani Gowda, and she's an assistant professor in Faculty of Health Sciences, Hokkaido University, Japan. So thank you very much, Dr. Divyavani, for kindly agreeing to deliver this month's talk. Now I request Lakshmi Pradeep to introduce the speaker.
Thank you, sir. Thank you very much for inviting me for this wonderful talk.
Dr. Divyavani Gowda is an assistant professor, Laboratory for Advanced Lipid Analysis, Faculty of Health Sciences, Hokkaido University, Japan. She obtained her PhD in Environmental Science from Hokkaido University, specializing in atmospheric aerosol chemistry. Her research focuses on air pollution, environmental chemistry, metabolomics, and advanced lipid analysis. She has extensive expertise in GC, GCMS, and LCMS techniques for environmental and biological sample analysis. Before joining the faculty, she held research positions at Hokkaido University and Rinken, Japan. Dr. Gowda has successfully led several CAF-ND-funded research projects on atmospheric pollutants and human health. Her work explores the formation, transformation, and health impacts of airborne organic compounds and PM2.5 aerosols. She has received several awards and is recognized for her interdisciplinary research contributions in environmental science and public health. Dr. Gowda actively collaborates with researchers worldwide to advance sustainable solutions for environmental and health challenges. Today's talk is titled Photochemical Formation and Transformation of Water-Soluble Organic Aerosols Revealed by Hydroxy and Keto-Dicarboxylic Acids in Remote Marine Aerosols. The talk discusses the seasonal distribution, sources, and photochemical formation of hydroxy and keto-dicarboxylic acids in total suspended particulate aerosols collected weekly over 1 year at a remote marine site in Japan. The study provides new insights into the mechanisms governing secondary organic aerosol formation and their implications for aerosol hygroscopicity, cloud formation, and climate interactions. We welcome you, ma'am, for the talk.
Thank you, ma'am. Thank you very much for the nice introduction. So I would like to start my presentation. Am I audible?
Yes, you are audible.
Yeah, okay. Okay, sir. I'll begin my presentation. Today I'm going to talk on photochemical formation of water-soluble organic aerosols and the formation of hydroxy and keto-dicarboxylic acids in remote marine aerosol. Before I move on to my actual topic, I'm going to explain my content of today's talk. So today I'm going to talk on the introduction to our Hokkaido University and the scholarship opportunities in Japan and the marine atmospheric aerosols, urban atmospheric aerosols. And addition to that, I'm also going to talk on air quality management in Japan and lipidomics based research at Hokkaido University. So let's move on to our university. Okay. So I belong to Hokkaido University. It is located in the Hokkaido Island. You can see this is the island, or this is the map of Japan, and our Hokkaido Island is located in north part of Japan. It's almost like an for India, Jammu Kashmir. So for Hokkaido is in head of Japan. So our Hokkaido University is located in the north part of island. And I think you all heard about the news yesterday. There was a lot of earthquake happened. So this earthquake was happened in the Fukuoka prefecture. So this was the earthquake affected area, almost like a south part of Japan. And this is the overall map of our university, Hokkaido University, and my faculty, Faculty of Health Science, is located in the center of our university. And our university was founded in 1876, initially as an Sapporo Agriculture College. Later on, it was named as an Hokkaido University. And until now, our university has received two Nobel Prize, one for the chemistry, both of our for the chemistry actually to be say. Hakiko Suzuki won the Nobel Prize in 2010 for the Suzuki coupling reaction, and the Benjamin List, actually he is a German professor. He is affiliated to our university, so he got a Nobel Prize for the asymmetric organic catalytic. So we are very proud to say that our university have received two Nobel Prize until now. And our university is located in the city center. So it's a very beautiful city to visit, and our university is full of a lot of greeneries. And he is the founder of our university, to say, Dr. William Smith Clark. So he is an American educator. Initially, he was invited into our university to teach English, chemistry, zoology, and botany. And later on, he is the one who changed the structure of our university after he visited our university. So still, we have his name in the many places of our university, like Clark Center or Clark Memorial room, something like that. So still we remember him a lot. And when we move next, I would like to talk on scholarship opportunities in Japan. So there are many scholarship opportunity for international students. First one is the Japanese government mixed scholarship. This scholarship can be obtained for the undergraduate, master, or the PhD student, even for the researcher. And there are a lot of beneficial in this scholarship. The scholarship will be fully funded, and the student who are selected get the round trip, and their monthly expense will be also provided. This scholarship has like a two-way. One is from the embassy recommendation, another one is from the university recommendation. This is the very prestigious scholarship. Addition to that, there is one more scholarship called JASO. This is for the international students who are already enrolled, like they are private financially funded students. They already enrolled the university. After enrollment, if they want to apply for some scholarship, they can apply for this JASO scholarship. Even though the funding is small, addition to this, even they can also get some funding from the professor. This is also one of the prestigious scholarship. And our university also have some other of funded scholarship can also be obtained. Addition to that, in most of the professors' case, he will be having, or she will be having their own project. Even professors can hire the PhD or the master students based on that project, and he can also provide the TA and RA money from his own grant. Even when I joined as a PhD student, I was funded from my professor grant. So my RA money I used to get from my professor funding. Addition to that, I got a JASO scholarship. And why we need to consider to move to Japan as an Indian student? Because according to Embassy of India, until now, as of August 2025, there are only 3,400 Indian students are there in overall Japan. So when you compare to other countries like China, they have like 1,23,000, and it very surprisingly, Nepal students are around 64,000. When we compare our Indian population and the Nepal population, we can clearly make out that Nepal students are too many in Japan. Why not Indian students are focusing entering into the Japan? Maybe because of the language issue, or they are not aware of the scholarship or university information. So I would like to invite all the professors and some of the students who are really interested to enter Japan. So please go through the scholarship website and you can easily get into Japan. And according to the Japan government, now they are planning to increase the Indian students, like they are planning to increase until 15,000 Indian students. They are increasing the scholarship number. So it's a great opportunity for Indians, researcher or the PhD students to enter Japan. And one more thing, entering Japan as a PhD student is much more easier than entering Japan as a postdoc or the staff member. So do not miss the opportunity as a students to enter Japan. So this is what I wanted to talk on scholarship. So next, regarding our university have two type of study program. One is like English one courses, and some are in the Japanese course. In the case of English, as this is the engineering related one, I'm just focusing engineering, global environmental management, and the environmental health science are and the chemical and material science. There are a lot, few more. One there this graduate school will be taught in English, and the most of the other graduate school are taught in Japanese. Even though I graduated from environmental science, my PhD, but still, even though it was in the Japanese, but it's not mandatory to know Japanese to be enrolled as a PhD student. Even for the day-to-day life inside the university, you no need to know so much of Japanese. But when you go out of the university for the normal communication, you need a little bit of your Japanese skills should be better. So currently I'm working in the health science. Both my PhD and my current research both belongs to Japanese category. So moving into actual presentation. So as we all know, our atmosphere is made up of primarily of gases like nitrogen, oxygen, argon, and the carbon dioxide, and some other rare gases. Addition to gases, our atmosphere also consists of particulate matter, like what we call it as a small liquid droplet or the solid particles. This particulate matter play a very important role for the human health and even for the climate change. When the atmosphere is very clean and clear, we cannot make out this particulate matter in our naked eye. But when it is highly polluted, as shown in this picture, we can clearly make out the existence of this particulate matter. This particulate matter and the aerosols seems they are similar, but when the definition is a bit different. An aerosol is a suspension of fine solid particles or liquid droplets in air or any other gases. So this is how we can define the aerosols. These aerosols have majorly two effects. One is we can consider as an indirect effect. Another one we can consider as a direct effect. Direct effect is related to health. In the case of indirect effect, these aerosols can affect like radiation, climate, remote sensing, and even the agriculture. So generally in the case of particulate matter, we are focusing on PM 2.5 and the PM 10. So I think you all know what is the difference between PM 2.5 and the PM 10. The PM 2.5 is the fine inhalable particle, and whereas in the PM10 is also inhalable particle, but these two are recently many people are concentrating more because they can enter human body very easily and they can cause a lot of health effect. And when we move into the composition of the aerosol, generally aerosols can be categorized into two major classification. One is inorganic aerosols, another one is organic aerosols. The aerosol that is made up of solid sodium chloride, ammonium sulfate, or insoluble minerals are considered as an inorganic aerosols. In this also there are two classification. One is primary inorganic aerosol and another one is secondary inorganic aerosol. Primary is nothing but that are directly emitted into the atmosphere, and the secondary inorganic aerosols are nothing but they are undergoing transformation in the atmosphere. In addition to that, we do have an organic aerosol, that is the hot topic of today's presentation. These organic aerosols are made up of carbonaceous particles like polycyclic aromatic hydrocarbons and the aromatic hydrocarbons. Here also there are two major classification, that is primary organic aerosols. They are directly emitted from the sources. It might be a natural sources or the anthropogenic sources. And whereas in the case of secondary, these are they are formed in the atmosphere. As we all know, the organic aerosols that are formed in the atmosphere are more dangerous for the human health than the primary one. The primary one are the hydrocarbon, pH, and the fatty acids are the example for the primary organic aerosol. Whereas in the case of secondary, we can consider as the dicarboxylic acids or its derivatives. And here is the cycle of aerosols. Aerosols are emitted from the various sources, and they are generally transported from one place to another place. And during the transportation, they undergo a lot of chemical transformation, and they do also interact with the clouds, has a cloud condensation nuclei or ice nuclei, and so on. And these aerosols can be removed by the wet or the dry deposition. Wet deposition is nothing but the snowfall or the rainfall. Dry deposition is nothing but the gravitational settling. This is how aerosol cycles can be observed. And today's topic will be the marine atmospheric aerosol. So mainly I'm going to talk on composition and the characterization. Why we need to focus more on organic aerosols? Because organic aerosols have an hygroscopic nature, and they also act as a cloud condensation nuclei. In organic aerosols, the dicarboxylic acids are the major water soluble fraction. It comes around 50 to 60% of the organic aerosols will be composed of a dicarboxylic acid. And when it comes to the total carbon, 16% of the marine atmosphere will consist of the dicarboxylic acid. These dicarboxylic acids have an huge impacts based on their chemical property. They are highly water soluble, and they have the low vapor pressure, and they are highly reactive in the atmosphere. When we look into their physical role, they involve in the new particle formation, and they also act as in cloud condensation nuclei, and also they play a very important role in the climate change. When we look into the health effect, these dicarboxylic acids are in the atmospheric aerosol contribute to the health risk, mainly like involvement in the PM 2.5. And they also, when we inhale this dicarboxylic acid, they act as an oxidative stress, and we also have an inflammatory response. Addition to that, this dicarboxylic acid are often indicated as an atmospheric aging. So quantifying this dicarboxylic acid will be a very important factor. So the major emission of this dicarboxylic acid will be from the automobile emission, fossil fuel combustion, and the meat cooking and the biomass burning. Addition to that, there are secondary sources like photochemical oxidation of the volatile organic compounds in the atmosphere. So addition to this dicarboxylic acid, some dicarboxylic acid consists of hydroxy and the carboxylic functional groups. They are considered as an hydroxy and the keto-dicarboxylic acid. This keto and hydroxy dicarboxylic acid are assumed to be a late product of photooxidation of unsaturated fatty acids or the cyclic alkanes. But there are not many literature evidence for the existence of this hydroxy and the keto-dicarboxylic acids in the atmosphere. So I focus mainly on the quantification of this hydroxy and keto-dicarboxylic acids in the marine atmosphere. This is the structure of hydroxy dicarboxylic acid, and this is the structure of the keto-dicarboxylic acid. As they have the additional functional group compared to the dicarboxylic acid, they are very active molecules than the dicarboxylic acids. So this is the overall pathway of the dicarboxylic acids. Once they are emitted from the source in the gaseous form, they can easily convert, they undergo transformation and in the aqueous form into the smaller molecules. For example, unsaturated fatty acid, it will be emitted in the gaseous state, but finally it will reach to the aqueous phase, and it undergoes smaller fragmentation and like a smaller formation, and finally it will leads to the oxalic acid. And the main objective of this study was to identify the homologous series of hydroxy and the keto-dicarboxylic acids in the marine atmospheric aerosol. And addition to that, we also tried to focus on the seasonal variation of this dicarboxylic acid and also we thought of studying the photochemical pathway of the production of dicarboxylic acids and in its intermediate products in the atmospheric aerosol that are emitted from the anthropogenic and the biogenic sources. And for this purpose, we collected the sample from the very remote island called Chichijima Island. This island is located here. You can see in this map. It's a bit far from Japan. And not many industry are located in this area. And it's purely natural. So the aerosol samples were collected from December 2010 to November 2012. We collected the total suspended particles on the weekly basis. Totally, we had around 53 aerosol samples. And when we look into the air mass back trajectory for the 7 days, what we noticed is in the case of winter is nothing but the December, January, and the February. The air mass are directly coming from the continent. Whereas in the case of spring, that is March, April, and May, it's influenced by the continent and even from the marine. It's like a mixed one. In the case of summer is June, July, and August, mainly influenced by the marine region. Whereas in the case of autumn, this area was mainly influenced by the transition. We have some air mass from the continent, some are from the marine. So this is a very unique sampling site. It has an source of aerosols or particulate matter from the continental or even from the marine region. So this is the seasonal variation of hydroxy and keto-dicarboxylic acid over the Chichijima, what I identify. What I noticed, this hydroxy and keto-dicarboxylic acids are very abundant in the spring region. The green color indicates the spring season, followed by the winter. Winter is the red one, and the autumn and the summer. So in which the malic acid, that is a C4 carbon atom, was the most abundant species in the detected one, followed by the 2-hydroxyglutaric acid is the second abundant hydroxy dicarboxylic acid what we detected in these samples. So when we compare with the dicarboxylic acid and the hydroxy dicarboxylic acid, the trend is similar. So in the case of dicarboxylic acid also, their concentration is very high in spring, followed by the winter. Even our hydroxy dicarboxylic acid have the similar trend to the dicarboxylic acid. So this clearly indicate that the sources of the dicarboxylic acid and this hydroxy dicarboxylic acid may be similar. This hydroxy dicarboxylic acid might be forming in the atmosphere from its precursor, that is dicarboxylic acid. And we also try to look into their comparison by molecular distribution between the oxalic acid. Oxalic acid is the final end product of the dicarboxylic acid. So this is the scale of oxalic acid. Here same. And this is the scale of the malic acid and the petonic acid. This is the hydroxy dicarboxylic acid. This is the structure of keto-dicarboxylic acid. Both hydroxy and the keto-dicarboxylic acid follow the same trend of the diacid, that is the oxalic acid. So this similar pattern clearly indicate that they are coming from the similar source and their formation pathway might be similar. So we also try to monthly variation of this dicarboxylic acid and hydroxy dicarboxylic acid with some specific tracer. That is the non-sea salt sodium and the non-sea salt calcium, and we also try to correlate with the MSA. So what we found in the winter and the spring, as we know, it's coming from the continental, whereas in the summer, this is the precisely only the marine air mass is coming for this Chichijima Island. This hydroxy and the keto-dicarboxylic acid show the similar variation with the anthropogenic and the biomass burning specific trace. So this clearly indicates the air masses that are reaching the Chichijima Island from December to April are suggested to be originated from the anthropogenic pollutant, whereas the minimum concentration observed from the May to August are due to the marine biomass. So this is, we can clearly make out based on the specific trace comparison. So this is the result of that. Addition to that, we also try to compare with the anthropogenic or the biogenic one. What we noticed, as the concentration of total hydroxy keto-dicarboxylic acid increases, the ratio also increases in winter, spring, and summer. You can see here it's increasing in winter, spring, and in the autumn. So this suggests that the anthropogenic origin is the main sources of this dicarboxylic acid formation in the Chichijima Island. Whereas in the summer case, this trend is bit negative. So this clearly indicates the aerosols are from the biological sources. This is the result of Chichijima Island. So let's move on to our urban atmospheric aerosol, lipid composition and characterization. So lipidomics of atmospheric aerosols are very much important because in the urban area, the atmospheric procedure like processing and the crucial for the development of air quality management and addressing their importance of human health is very much important. Lipids are play a very important role. Sorry. Lipids are very important because they are hydrophobic components of organic matters. However, their presence, composition, and atmospheric role remain purely and it's not been estimated. So the main objectives of this study was to analyze the lipid composition of atmospheric aerosols that are collected in saporosity and to investigate the seasonal variation in lipid level in atmospheric aerosol, identification and characterization of previously undetermined lipids in aerosols. So for this purpose, we collected the aerosol sample from October 2021 to September 2022 at the Department of Health Science, Hokkaido University. Upon collection of this sample, all the samples were stored at minus 80 degrees Celsius until further analysis because lipids are very sensitive. If the temperature is bit high, their composition will be changed. So we stored the samples at minus 80 degrees. So this is the quartz fiber filter where we collected our aerosol sample. This is our ambient air sampler. And we can even monitor our airflow. So this is the protocol what we follow to extract the lipids from the aerosol samples. Generally, we cut 32 centimeter of diameter of this filter and we add the internal standard and we use the chloroform and methanol as the solvent for the extraction. And then finally, we injected into the LCMS. What we noticed here is many fatty acids are obtained in high concentration in the spring. As you know, our Hokkaido is highly, it's like a cold area. In the case of winter, we received a lot of snowfall. So almost almost no leaf in winter, to be frank. So when the spring started, we will notice the small leaves, and by the end of spring we get everything will be greenery. So that's why we can see lot of fatty acids in the spring season. So the mainly the palmitic acid, that is 16:0, is the most abundant fatty acid what we notice, and fatty acid 12:0 was relatively higher during the winter season. Probably this fatty acid might be coming from the combustion source because of the cold weather. We use lot of heaters here. So probably this fatty acid 12 might be obtaining from the combustion source. And the fatty acids are found to be contribute to 5 to 55% of compounds identified in the meat cooking and automobile biomass burning and on the leaf surface of the urban atmosphere. These are the various fatty acid what we have identified and quantified in Sapporo aerosols. And very interestingly, we also try to look into the composition of these fatty acids in different season, like a monounsaturated fatty acid and a polyunsaturated fatty acid and a saturated fatty acid. But we didn't find any clear seasonal variation between saturated and the monounsaturated fatty acid. But can you hear me? Hello?
Yes, yes, we can hear you.
Oh, okay. Sorry, I heard something. Okay.
No, no.
The
Right.
FUFA, yeah, sorry. The FUFA, we noticed the very clear seasonal variation between the spring to autumn and spring to winter. So in the case of saturated fatty acid, the carbon chains are straight without any double bond. Whereas in the case of polyunsaturated fatty acid, what we can see, there are some double bonds in the carbon chains. That's why it is called as the polyunsaturated fatty acids. In the case of monounsaturated fatty acid, if there is a single double bond, it is called as a monounsaturated fatty acids. So this is the seasonal variation of the fatty acids that have been obtained. So addition to that, we also identify the phosphatidyl lipids, like a phospholipids. phosphatidylinositol. So these phospholipids are higher in the spring compared to other season. And this also have the similar trend to the fatty acids. These phospholipids are present in the atmospheric aerosol, particularly in the marine aerosol and in the biological sources. And they are typically low in abundance, but are environmentally, they have a lot of importance because they influence the cloud formation. They also influence in the atmospheric chemistry, and they also influence in the biomarker tracing. So this heat map clearly shows that their concentration is very high in the spring season, whereas they are almost like in the under detection limit in other seasons. So other lipids are also identified, like sterol lipids. Sterols are widely detected in atmospheric aerosols generally, and their origin from the plant, fungi, animal, and the marine origin and the biomass burning were also being traced. And the very unique lipids have been identified, that is the FAFA. So this is the structure of FAFAs. This FAFA is nothing but fatty acid esters of hydroxy fatty acids. These are relatively, like in 2003, these fatty acids have been discovered in the human adipose tissue. So until now, lot of biological experiments are going on to know what is the biological function of these FAFAs. But this is the first time to identify these FAFAs in the atmospheric aerosol samples. So there are not many reports have been identified or quantified. What are the sources of these FAFAs and how these FAFAs are forming in the atmosphere, and what are their effects on the human health, or what of their effect on the climate change is unknown. So we need to be focus more on this type of FAFAs. So this is one more study. And let's move on to another one is the air quality management in Japan. So I'm the chemist, so I just until now I just focus on the chemistry of the aerosol and the air quality. So I just try to grab some information because I'm in Japan from last 13 years. So I know the air quality in Japan, it's very good. So I really wanted to speak something about the Japan to my audience. So I grabbed this information. I'm not sure about this because I got it from the Google, and I used the help. I took the help of ChatGPT to make these slides. So please forgive me if something is wrong. So air quality measures are very serious in Japan because they have the very strict standards of air quality measurement. Generally, they have the annual mean of PM 2.5, that is less than 15 microgram per meter cube. And in the case of PM10, it's around 100. And they do have a monitoring for nitrous oxide, sulfur dioxide, and the carbon monoxide, and even for the photooxidation, that is ozone. And this is very interesting for me. You know, there are more than 1,500 ambient monitoring sites throughout Japan. And whereas in the case of India, I'm not sure how many are there. If we check the area of Japan and the India, we know India is so big. So if we calculate, we should have around 13,000 monitoring sites. I'm not sure whether do we have this much? Hello?
Yes, go ahead. Actually, somebody might have been unmuted it. Yeah, please, all of you others, please unmute your mute your mic.
So in Japan, we have more than 1,500 stations. So in this station, it's some are located in the roadside, some are located in the rural background, some are located in the industrial area. So all this monitoring site will measure the PM 2.5, PM10, and the nitrous oxide, sulfur dioxide, carbon monoxide, ozone, even volatile organic compound, and the black carbon will also be measured. And it is a real-time data flow, so we can clearly check this data. So addition to that, in Japan, we have a very clear vehicle emission regulation and industrial emission control. When it comes to vehicular emission, we call it as an SAKEN, that is called the mandatory vehicle inspection. All the vehicle need to be inspected every 2 years. Even though if you buy new vehicle from the showroom, you need to take to this mandatory inspection location, and you need to check your newly released model, get the certification from this inspection, and you need to put your certificate on the car. So then it will be like a circular one. So it includes the date and the month of your inspection. So after 2 years, you need to go again and get the new one. Once the inspection is done, you need to replace with the new one. So this is a very serious inspection will be done for all the vehicles. During that inspection, what they will do, they'll check the carbon monoxide emission from our car and other one. Addition to the emission, they also check the light and the indicator, and even the wipers will be checked. So even those things are very much important when we are driving our vehicles on the road. So those things will be also checked in this mandatory vehicle inspection. Addition to that, they also have the low sulfur fuel, so that will be monitored. And they also promoting for the hybrid, the electric vehicles, and they also use the catalytic converter. And when it comes to the industrial emission control, they have the very strict rules and regulation for the industries. They control the fuel gas desulfurization, and they have the selective catalytic reactor. They do have an electrostatic precipitator, and they do have a fiber bag to collect the particles before releasing the gas into the atmosphere. So these rules are very strict, and all the industries, even the vehicle, will be followed. So that's why the air quality in Japan is very clean, and all the public area will be very protected. And they do have a monitoring for the transportation of the air pollutant from the cross boundary, because Japan is located in the east part. So the wind from the China and the Mongolia is very strong in the spring season. So before they noticed, they even informed to the public that the like what they call red sand or the yellow sand will be visiting us, so please be careful, something like that. We often we receive the message from the pollution control board. And they do have a promotion for the clean energy, like renewable energy. Hydrogen technology has been invented. So a lot of things have been, I know this all also implemented in India, but not visible. But in Japan, all these are implemented, and it's visible to the public, not just the researcher. So these are the air quality measures that Japan has been taken. In the case of urbanization, generally most of the people use public transport. Yes, of course, Japan has a very good public transportation when we compare to India. So even though they have the car, they use the public transportation, and most of the people use the bicycle. And we have a lot of greeneries, and the low emission zone will be also considered. So these are the air quality measures that has been taken in the Japan. So addition to that, I would like to speak few things about the lipidomics based research at Hokkaido University. Even though I did my PhD from environmental science, after my PhD, I switched my research field to lipidomics. This is possible in Japan. If you are interested in another field, you can easily switch. It's not a big issue. So why we need to consider about the lipids? Why lipids are important? We all know the macronutrients are the like carbs, protein, and fats are comes under the macronutrients. And we also know that all fats are lipid, but not all lipids are fat. Yes, of course. The lipids are the broad category of organic molecules that are insoluble in nature, whereas fat, these are only specific type of lipids that is generally comes under triglyceride, and these are insoluble in water, and they are solidified at the room temperature. So there is a very clear difference between the lipids and the fat. Most of the people will confuse the fat with the lipids. Why we need to consider more on lipids, I'm going to explain in detail. This lipidomics is recently, like in the 2003, it was separated from the metabolomics. Initially lipidomics was inside the metabolomics, but in the era of lipidomics was came into exist in the 2003, where the lipidomics become separation from the metabolomics. Until now, nearly 49,000 of lipids have been identified and categorized based on their structures. These lipids play a very important role. You know, all our cell are made up of lipids. The cell wall, even it is a plant cell or the animal cell, the cell wall is made up of lipids, and it's play a vital role in the signal transfer in and out of the lipids. If you look into our adipose tissue, human adipose tissue, we have around 64% of triglyceride, and the remaining all are the different type of lipids. So these lipids play a very important biological role, and they also act as an autoimmune disorder. They also play in the neurological one and the cancer, and they also act for the infectious diseases. So they play a very important role in our day-to-day life. So consuming the lipids is important, but we need to be consider it should be in a moderate, and what type of lipid we are consuming, it's also play a very important role. And we know in the case of lipids, there are two type of lipids, that is exogenic and the endogenic lipids. Exogenic is nothing but the lipids that need to be consumed in our food, like omega-3, omega-6, many times you will be known. Whereas the endogenic lipids are the lipids that our body will synthesize. So we no need to consider these lipids to be consumed. So there is a very clear differentiation between the exogenic and the endogenic lipids. Our lab is mainly a laboratory for advanced lipid analysis. What we do, we chemically we synthesize the lipids and we isolate the bioactive lipids, and we do the lipidomics, like food lipidomics and the clinical lipidomics. Even we get some clinical samples like urine sample, plasma sample, even some tissue, brain tissue, heart tissue, kidney tissue. And people, other scientists will ask us to compare the lipid composition between the healthy person and the diseased person. These lipids, if their concentrations are in the balance, it's very good. For we are healthy. If the balance is imbalanced, like if the lipids concentration are down or if the lipid concentration are bit high, it's very bad to our health. So that's why many clinical scientists, they send us the sample to know the lipid concentration. Addition to that, we also focus on the functional lipids, that identification of functional lipids in the different type of food. So we focus on the herbal tea, we focus on beans, different type of seafood, even the grains like rice, sorghum, and even some rose. These are the main research what we have done in our lab until now. So this is our team called, generally we call it as a LALA, Laboratory for Advanced Lipid Analysis. So she's our professor and the director of our LALA, Shu Ping Hui. And this is our lab associate professor, Dr. Gowda. And this is one more our associate professor, Professor Sakurai. And this is our assistant professor, Dr. Eddy, and here is one more our assistant professor, Dr. Permal, and here me, it's Divya. So this is what I'm going to talk on today. So thank you so much.
Thank you, Divya. It's an excellent talk. Yeah, I hope many people get some insight into the lipids, and particularly coming from the cleaner environment. So,
Yes, sir.
so before we take up the question, I'll request everybody to start your camera. We will have one, virtual image now, and we'll take a photo of that, and then after that we will discuss about the question answers. So, request everyone to start your camera, and if you have any questions, please, type in the chat box. Okay, please start your camera, all of you. Okay, maybe Divya, can you just unscreen your
Yeah, yes, sir.
stop
I'll
sharing
just
it?
stop it.
Done? Lakshmi, done? Photo?
Yes, sir.
Okay. Thank you very much. Divya, I mean, it's a very wonderful talk, and I could able to get more insight into the lipids and understand that, even though if we are thinking about addressing, air pollutants, vegetation would be a good, but that can also lead to some newer pollutants. That's an interesting to be look at it. So did you look at it? What kind of species? Whether sometimes no. In Indian context, some of the native species will be there. Sometimes migrated species will also be there. These migrated species will contribute some kind of these, toxic, chemicals that may be harmful. The native species maybe they're existing for quite long, and they in turn contributing to the overall development of the community or the environment in that particular area. So, do you think that did you have some idea about whether the different types of species will influence in terms of contributions to the lipids?
Yeah, I think it's might be true because different species have the different wax on their leaf. So if the wax is changed, the lipid composition will be changed, and even the wax composition will be depend upon the temperature.
Okay.
Okay, if the temperature is a bit high, the oxidation will happen. So oxidized lipids will be more.
Okay.
So in order to know which I saw one comment in the chat box, they are mentioning what type of plant species will produce what type of lipids. So in order to know that, we need to first analyze the plant species and compare with the aerosols. Then we can clearly correlate which type of plant species are generating what type of lipids. We are just an aerosol scientist. We just collect the aerosol samples and we measure the composition. If we can collaborate it with some biology people or the botany, if they send some samples, leaf samples, we can analyze.
Okay, that's interesting.
And yeah, I guess that's it.
So there's another. So you just mentioned about it. There are excellent monitoring network in Japan, and so. I mean, is there all manual monitoring station, or you also have some real-time monitoring station?
These are real-time monitoring station. What 1,500 are the real-time monitoring.
Oh, that's a really good large data.
Yeah,
sets.
very big number. Yes, very big number. And you can see in the map, it's covered throughout the Japan.
Okay. So is it they generally identify to set up these monitoring stations to location specific? For example, they wanted to monitor the industries pollutants or traffic based or residential based. How do they classify? Do you have some idea about it?
No, based on the what they are getting the information, based on that they are set up in the new one. If in the industrial area they set up in one place, and if the particles they are getting more and more, they are set up in one more, and within the 2 to 3 kilometers, and they are monitoring. If frequently if the data is a bit high, they are informing the industry to take care of those pollutants.
That's nice. Okay, so then we also all of them are continuous monitoring. Do you also have some manual monitoring stations?
Yes, we do have.
We do. Okay. Then there's another question you can go through if you can read it from your screen. There is a airborne
I think
pollen.
someone is rising, Dharmendra sir, Professor Dharmendra. We can take him quick call before I read the question. Yeah.
Yeah, go ahead.
Yeah, thank you. Actually, I would like to know one and two questions here. So as you said, we have to during collections in high volume sampler, that temperature is maintained minus 80 degree.
Yes,
In
sir.
high volume sampler, how you maintain this?
Not in the sampler. After collecting the sampler, we are storing the sample in the minus 80, so that it should not undergo further oxidation.
Okay. During transfer.
During the transfer, yes.
That we have to maintain. During the collection time, need not require.
The collection time, we cannot. What is happening in that area, we need to know until from this day to this day. So we collect all the sample aerosol particle and then we store.
Fine. And another question is there in Japan, there are some standard guideline for lipids?
No. So this is no, there are no much guidelines for the lipids, like the concentration of the lipids. We don't have anything.
Okay. Thank you for my question.
Thank you, sir. So someone mentioned we have around 17,000 monitoring site manuals in India. But 17,000, it's a really small number for India, I guess.
Yes, yeah, that's true. We are keep increasing the number of monitoring stations.
Yeah. As airborne pollution only in one of the serious problem. How is this problem being affected there and what are the available standards? Okay, in Japan, pollen allergy is a very big, serious issue. Yes, I do accept this. So it's difficult because the pollen are from the natural source. We need tree. We cannot cut down the tree for to avoid the pollen, right? So what people will do, they wear a mask and they try to stay indoor during the pollen season. The pollen season will be for short time. So generally the pollen from the sakura plant, and there are two more varieties of flowering plants. So only the pollen will be in the particular season. So people try to stay indoor, or they try to avoid the area where there are such trees with lot of pollens. So, but we don't have any particular uh, regulation for the control of the pollen because it's a natural source. And,
In thermal power plant, they have the fugitive gas, uh, device will be there. So is being used. What is the use of waste generated from the FGD? I think it's basically looking at in terms of fugitive gas desulfurization. So so do you have some? If there is a
Oh, no, sir, I don't have any much idea about this power plant AFDG. I'm sorry for this.
okay, then Government of India passed
Cabinet
in
Chemical Park. I need to work next to.
2004 for chemical
Oh.
park, and it will be work in next 3 to 4 years. Then after a lot of aerosols will produce. Yeah, I mean, let's see. I mean, it's a we don't know how this chemical park will contribute in terms of pollutants. Do you have some comments on that one?
Sir, having the chemical park is not an issue, but the thing is how the filter has been kept in those parks, it will play a very important role, right? So
Correct. Then
what's the status of bioaerosol studies in India?
oh
Sir,
yes.
do you have any idea about the bioaerosols in India? I don't
Yeah,
have
yes,
much
there are
idea
many
about this.
studies. Yeah, there are many studies going on. Even in our group, colleagues are working on bioresource measurement. And if you can visit our website, people can look at it. Shakil, maybe you can visit our website. The colleagues like Professor Sachin, Professor Chandan, and Professor Vikram, they're all working in bioresource. And very many faculties across in different IITs and IIC also working on this bioresource. Okay.
So
Would any
someone asked me one more question, sir. So is there are any chemical reaction is going to take place on the filter, and is there are any specific standards have been kept for lipid sampling? So first of all, for the filter, generally the quartz fiber filter it's not highly reactive. And before collecting the aerosol sample, the filter will be baked around 450 degrees Celsius for 6 hours. So all the carbon particles or some other particles that are present on the quartz fiber filter will be removed before we collecting the samples. And during the collection of the sample, yes, we kept the sampler for 1 week. During that time, if it is raining or if there are a lot of insects, even I notice sometimes some insects falling on my filter paper. So those things we cannot avoid this. It will be like normal for not always, but sometimes we can see some wet aerosol filter also. So there are not much standards have been placed until now for the lipid sample collection. And definitely ambient temperature play a great role in collecting the pollutant. What average ambient temperature in Japan? Yes, ambient temperature play a very important role when it comes to aerosol. Even the geographical location of the uh, sample collector will also play an important role, because in the case of Japan, Osaka city is the most polluted city in Japan. The main reason most of the industrialization from the Edo period was taken place in Japan, nearby the Osaka city, Osaka or the Kyoto city. So that's why from the Edo period until now, Osaka is considered as the most polluted city of Japan. And addition to that, if you see in the map of Japan, Osaka is in the like a center, south to center part. So this is very close to China. So cross-boundary air pollutants can easily transfer to Osaka region. So that's why it also has an impact to get the more PM 2.5 or the PM10 in the Osaka area. So we can say yes, ambient temperature also play a very important role for the aerosols.
Okay,
So
there are no other questions. Thank you, Divya, for excellent talk and clarifying many questions. As
no,
this is
thank you.
a new area which you are working on, air pollution and lipids definitely will going to bring some new insight into the health risk assessment. Thank you very much on behalf of Air Quality Management lecture series and all colleagues, and I would like to thank you for in a short notice agreed and accepting our invitation and delivering this wonderful talk. So we will also looking forward to all participants for this air quality management lecture series in the next month lecture. Until that, bye to all of you, and thank you so much. Thank you, Divya.
Thank you, sir. Thank you so much for inviting me. Thank you so much. Bye bye, sir. See you.
Thank you. Bye bye. So
Thank you,
bye bye
sir.
to everyone. See you.